This co-culture method will help answer key questions surrounding the cell contact mechanisms behind mesenchymal stem cell regulation of macrophage phagocytosis, and therefore illuminate the role of MSC in regulating innate immunity.
This technique can be applied in high-throughput analyses. The bioparticles conjugated to pH-sensitive dyes fluoresce only in acidic phagolysosome environments. Hence, washing and quenching are necessary.
This method can also be applied to a variety of co-culture models that include neutrophils and other phagocytes.
Demonstrating the procedure will be Ethan Chetkoff, an undergraduate currently in my lab.
To begin with, observe the confluency of cultured mesenchymal stem cells. When 70 to 80% confluency is achieved, aspirate the growth medium from the cultured cells in a 100 mm dish and add 5 ml of PBS. After aspirating the PBS, add 2 ml of 0.05% trypsin EDTA solution and incubate the cells for 3 minutes. After 3 minutes, check the cell detachment using an inverted microscope, and if the cells are not detached, incubate for 1 to 2 minutes again. Once all the cells are detached, add 5 ml of fresh growth medium to the plate. After rinsing the plate using the trypsin medium mixture, use a sterile serological pipette to collect cells into a clean, sterile 50 ml conical tube. Next, pellet down the cells by centrifugation. After aspirating the supernatant, resuspend the cell pellet in 10 ml of fresh growth medium by gently pipetting up and down. For cell counting, add 10 microliters of cell suspension into a